I'm going to give a talk about frontotemporal dementia, outlining the broad landscape of the condition, talking a little bit about clinical presentations, pathology, genetics, and outlining some of the work that we do at NeuRA on FTD. Here we have NeuRA. So our research is focused on frontotemporal dementia and motor neuron disease, which, as I'll explain, are overlapping conditions, looking at multiple aspects of this, ranging from genetic and pathological through to the impact of the disease on caregivers and the burden, to inform us about the disease, but also to inform us about normal cognition from the way that it breaks down in frontotemporal dementia, which offers a unique window into cognitive organization. Now, what is frontotemporal dementia? Well, this is the disease that used to be called Pick's disease, but the term Pick's disease fell into disuse and was applied to a very specific pathology, so the term frontotemporal dementia replaced it. And as we've learned in the last few years, there's considerable overlap between FTD and motor neuron disease, and I'll talk a little bit more about that. There's also overlap between the disorders that we study and the complex movement disorders, such as corticobasal degeneration. Now, when we see a patient clinically, we classify them based upon their clinical pattern, how they come to us, and we divide the patients into those with predominantly behavioral changes, alterations in personality, social cognition as we call it, or changes in language ability. But often there's a degree of overlap, and individual patients can have both of those. Within the language patients, which is also called primary progressive aphasia, we further subdivide them into the pattern that they present with. So we have a clinical classification of cases that come along to the clinic and present to us. But when these patients die, the pathology is quite diverse. Unlike when we say somebody has Alzheimer's disease in life, we're very good now at predicting that they will have a specific pathology, plaques and tangles. But when somebody with FTD dies, they may have a number of different pathologies. They may have tau inclusion disease. These are the bodies that became known as Pick cells and Pick bodies. But there are a number of other variants on the tau pathology as well. Even within tau, it doesn't appear to be a single disorder. And then it was discovered a few years ago now that there was an entirely different protein that could be found in patients with FTD. This protein is called TDP, tau DNA binding protein, and actually appears to constitute the majority of cases. And within the TDP, there are a number of subforms of TDP as well, depending on which cells are involved. And it's interesting that tau is a microtubular binding protein, so it's to do with cell axonal transportation, whereas the TDP is a DNA RNA carrier protein. So they have very different roles in the cells, yet manifest in a different way. And then there's a small proportion of cases with yet another protein known as FUS. So obviously the drug therapies now are developing to target pathology, clearing the sort of pathology from the brain. But how do we know which pathology our patients have when they present with one of these syndromes? So a central aspect of our research has become, how can we predict the pathology? And can we do that from the clinical pattern? Well, we can to some degree, as I'll tell you, Or in the difficult cases, are there other things we can do? Detecting proteins in the blood, so-called biomarkers that may help us, or sophisticated forms of scanning to detect these proteins in the brain. So these are some of the current research directions. We also know that FTD is quite a familial or inherited disorder. If we dig around in the family history, we find that up to a third of cases, there is some family history of dementia or of motor neuron disease in a family member. And now, in families with a strong family history, we can detect a gene defect in a high proportion, certainly over 50%. This just shows our series of cases. We had seen 307 patients and collected detailed family histories in all these individuals. And the rate of family history in FTD was considerably higher. These are patients with a strong family history. Whereas the Alzheimer patients that we see of the same age, it was only around 5%, and controls similarly a small number. But if we look according to those clinical syndromes that I showed you in the first slide, the strong family history isn't distributed across the clinical subtypes. The patients with a strong family history tend to present with the behavioural variant, BV FTD. So if we see somebody with behavioural variant FTD, we're particularly keen on unearthing any family history that's relevant. Whereas the language presenting cases, who are the next two bars, they have a much lower rate of family history. And if patients present with the combination of FTD and motor neuron disease, labelled ALS here, then again they have a strong family history. So the rate of high family history isn't evenly distributed across the clinical subtypes. Now, there have been a lot of developments in gene identification. There are a number of genes that can cause FTD. Some of them are extremely rare, like the valsine-containing protein. The first two that were discovered are at the bottom. You can have defects in the gene that codes for tau. So some of the tau cases have a positive gene mutation. That turns out to be relatively unusual. The next gene that was discovered is on the same chromosome, labelled as progranulin. Patients with the progranulin mutation get TDP pathology, and again accounts for another proportion. But we knew there are a lot of cases with a positive family history, and particularly the families in which motor neuron disease and FTD cluster together. There was known to be a gene on chromosome 9, and then 2 or 3 years ago that gene was discovered, which has the rather unmemorable name of C9orf72, because it's on chromosome 9, and that describes the position it's in. Now, this turns out to be the commonest genetic defect responsible for FTD and motor neuron disease. So amongst those patients with a strong family history of FTD or of a mixture of FTD and motor neuron disease, a lot of them have this gene defect. And it's turning out that a number of the patients that we see who don't have an apparent family history, so-called sporadic cases, also have this gene defect, particularly those with the behavioural presentation. And some of the family members of those patients have been diagnosed with psychiatric illnesses, because we know there's an association between this gene and psychosis. The FTD patients with the C9 mutation have a high rate of psychotic behaviour. So another clue to us is when we see a patient with FTD, they've had psychotic features at some stage, they may have other family members who've been diagnosed with psychosis, and we find this gene defect. And some of these patients present rather atypically with a long, slow, indolent course. So I've talked a little bit about the clinical presentations. I've said a bit about the pathology and the genetics. I'll attempt to bring these things together. Now, when we see patients for assessment, many patients that we see have been, someone has thought they have FTD. That's not always correct. And the assessments that we put patients through when they attend here is we give careful attention to behavioural symptoms, changes in personality and social cognition, which involves mainly talking to the families. We're interested in how the patients are functioning and the impact in everyday life, because that's important for prognosis and the impact on the caregivers and the need for support. There's neuropsychological testing that helps us to diagnose these syndromes. There's brain imaging of different types, structural and functional brain imaging, and we're interested in blood markers, but that's in the sort of research domain. Now, the commonest way for patients to present is with the behavioural variant of FTD. And there are a number of symptoms of this which have been refined in the last few years, and we've been part of the international consensus on criteria for FTD. And there are a number of symptoms, but the key ones are disinhibited and socially inappropriate behaviour, losing regard for the normal social rules of society, becoming impulsive, rash, careless. A loss of empathy and sympathy and care for others. Strange, perseverative, repetitive patterns of behaviour. Changes in appetite and food intake, with overconsumption of food and craving sweet food. Now, I've highlighted these in red because we believe these are the kind of core distinguishing clinical symptoms, and we've been interested in studying each of these, attempting to come up with better tests so we don't have to rely entirely on what the families tell us, tests of disinhibition, tests of empathy and emotional judgment, and measuring food intake and looking at people's appetite. And we've been looking at the areas of the brain that are responsible for the genesis of these symptoms. And when we see patients, we diagnose someone as having possible FTD if they have 3 of these, but there's no abnormalities that we can see in the brain, there's no shrinkage of the brain. And we put them into a probable category when there are these symptoms plus imaging abnormalities, and there's obvious evidence that this has got worse. We do this because there's considerable overlap between these symptoms and psychiatric disorders, and so not everybody with these symptoms evolves into FTD. And we use a range of instruments which can be found on our website. So I've talked about these key behaviours, and we've been mapping these to areas of the brain. And most of them you can relate to key structures in the brain that are shrinking early in FTD. And these areas are on the inner part of the frontal lobe of the brain, which is very important for inhibitory control. And the other area that's shown lower down in orange on the right-hand upper panel, it's called the amygdala, and that's very important for emotion. Other parts of this circuit are very important for appetite control. So this circuit is involved in BVFTD. Now, when patients present with behavioural variant FTD and they die, a proportion of them have the C9 mutation, but pathologically they can have either of these pathologies. And we're not very good at the moment of saying which patient has which pathology. So again, a sort of research development in the next few years is identifying better markers, whether they're imaging or whether they're blood markers of the underlying pathology, because the split is about 50-50. When we come on to the patients presenting with language problems, the situation's rather different. The language syndromes that we've delineated are more predictive of the underlying pathology. So these are patients that present with progressive impairment in communication, invariably difficulty with words, who are functioning otherwise quite normally in everyday life and do well on general tests of cognition. Now, these patients traditionally have split into semantic dementia, which is characterized by severe problems understanding the meaning of words. These patients complain that they no longer understand what words mean, they can't name things, but their speech is structurally normal and it sounds okay. It's not a problem of the organization of speech, it's a problem with the underlying meaning of that underpins language. These patients, as we've learned in the last few years, rarely have a positive family history, they don't show gene defects, and they have a specific underlying pattern of TDP. So it's a rather unique disorder in the way that it presents and the underlying pathology. The other syndrome, which is a disturbance more of speech and language production, in which speech is effortful and they grope for words and the grammatical structures of language are disturbed and speech sounds very abnormal and they produce errors. As patients died with this disorder, we discovered that the pathology can be quite variable, although a lot have tau pathology, unlike the semantic dementia. Quite a few of these patients actually have Alzheimer's disease in the brain. And in the last few years, we've begun to be able to distinguish those with Alzheimer's who are sort of catching us out as a sort of aphasic syndrome. We might think they're in the FTD category, but they actually have Alzheimer's. And they have rather different characteristics, which we've become good at detecting. And we came up based upon, we scanned these patients to know who had Alzheimer's in the brain using specific markers of Alzheimer's, and we looked at the clinical characteristics, and we found that analysis of three key aspects of language would distinguish these syndromes very well for us. And that maps onto very nicely the distribution in the brain. So the three syndromes of language disturbance, the semantic dementia, shown in the upper left panel, involves very much the anterior temporal lobes. The non-fluent patients who have disturbances of grammar and speech production, they have shrinkage. These maps show thinning of the cortex. So we can see thinning of the cortex in traditional areas to do with speech production, called Broca's area. And the Alzheimer patients presenting with aphasia, although they have a fairly pure aphasic syndrome, actually have very extensive brain thinning and pathology, and that progresses very quickly. Now, the final thing I'll talk about for a few moments is the overlap between motor neuron disease and FTD. Well, we knew at a clinical level that a proportion of patients with FTD develop motor neuron disease characterized by slurring of speech, difficulty swallowing, wasting of the muscles in the limbs. And also, a proportion of patients presenting with motor neuron disease develop FTD. And that became apparent gradually in the last sort of 20 years. But then the pathology in MND was discovered to overlap very considerably with the pathology in FTD. The pathology that I described, the TDP43, which underlies at least a half of FTD cases, is the same pathology that's found in motor neuron disease. So they share a pathology. And recently, the gene, the C9 gene, has been found in a lot of these overlap cases. So there's a clinical, pathological, and genetic link between FTD. So we have been very interested in this continuum between motor neuron disease and FTD. And in a nutshell, patients presenting to a motor disorder MND specialist and an MND clinic, about 15 or 20% of those will develop frank FTD symptoms. Usually the behavioural changes, sometimes the language changes as well. But a much higher proportion, getting up to a half of people with MND, have subtle changes in cognition and behaviour, judgment, motivation, emotional problems. And on the other side of the coin, again, of patients presenting with FTD that develop motor problems, a proportion have both FTD and frank MND, but we found through some research here that a much higher proportion have subtle features of motor neuron disease. So there's considerable overlap, and I've mentioned this mutation. So we mustn't forget that FTD causes very considerable burden and stress on families. This was some research we did a few years ago, looking at the degree of burden that family caregiver members are under. And the proportion of patients shown in black in the left-hand panel with a high degree of burden from living with somebody with FTD, virtually all of the family members were suffering enormously, much higher than in Alzheimer's and the language variant cases. And we showed on our rating scale, the FRS, which really rates functional ability, that as these patients progress, the burden increases. So the management of these patients requires, I think, a multidisciplinary team of the type that we're lucky to have here, with input from neurologists, advice on genetics, input from psychology, speech and language, occupational therapy, assessment, to see what can be done to help. To intervene with the carers who are desperate for information and knowledge. We've run carer intervention groups, which we show reduces their burden and depression. We advise on drugs for use in symptoms of FTD, controlling the disinhibition, for instance. And we're involved with trials of new agents to actually modify the disease. And of course, all this work is done by the team at NeuRA, who are shown here, and our team co-directed by myself and Olivier Piguet. So thank you very much.